cells, you need to know the organelles and other structures that can be found in animal and plant cells. An organelle is a specialized subunit within a cell that performs specific functions. All organelles found in animal cells can also be found in plant cells, so we will start with animal cells. Under a light microscope, you can see the cell membrane, nucleus, and cytoplasm. You will need to know their respective functions. Under an electron microscope, you can observe more organelles. The only tricky part is differentiating between the smooth ER and the Golgi body. Just remember that the smooth ER has connections between its layers, similar to the rough ER, while the Golgi body does not. You will also need to know the basic functions of the organelles. For mitochondria, you need to understand what the energy produced is used for. As for plant cells, under a light microscope, you can see a large central vacuole, chloroplasts, and a cell wall, all of which are absent in animal cells. These are their respective functions. Under an electron microscope, more organelles can be observed. Note that these are typical cells. Specialized cells differ in shape, and some organelles may be absent. Muscle cells have numerous mitochondria since they need to contract frequently. Root hair cells have elongated root hairs, giving them a large surface area. Their cell sap is concentrated, resulting in a lower water potential. They also lack chloroplasts. Red blood cells have several adaptive features that you need to be aware of. Next, it will be the movement of molecules in and out of cells. Diffusion is a passive process that doesn't require energy. You will have to know the definition, and the word net is important. It means that molecules move in both directions. There are just more molecules moving towards the direction with lower concentration, so they will be equal at the end of the day. Diffusion can occur in liquid or air and does not require a partially permeable membrane. Factors that affect the rate of diffusion are these four. The latter three are frequently applicable in questions on adaptation, while for size and mass, you should be aware that basic molecules can diffuse across a partially permeable membrane, while macromolecules cannot. Next, let's look at the net movement of the solvent molecules. Osmosis is the net movement of water molecules from higher water potential to lower water potential across a partially permeable membrane. It is important to take note that water potential is always negative. Hence, water molecules move from less negative, negative 300, to negative 600 pressure in this diagram. You will have to know the effect of osmosis on living cells. In animal cells, such as red blood cells, when the water potential of the surroundings is lower, water molecules will move out through osmosis, causing the cell to become spiky at the edges, and we say that the cell has crenated. While in a solution of higher water potential, water molecules will move in by osmosis, causing it to burst. For plant cells in a solution of lower water potential, water molecules will move out by osmosis, causing the cell to become flaccid. In a solution of even lower water potential, the cell membrane will tear away from the cell wall and the cell is said to have plasmolysed. In a solution of higher water potential, water molecules will move in by osmosis, and the cell becomes turgid instead of bursting as it has a cell wall. Note that when there is a change in the mass or length of a plant sample, it is always due to the movement of water molecules, and so you must always use the water potential gradient as a comparison rather than the concentration gradient. Sometimes, cells need to move molecules against a concentration gradient, and this requires active transport. Active transport requires energy from respiration to move molecules from lower concentration to a region of higher concentration. A partially permeable membrane is always required. This occurs in places such as root hair cells, where ions are pumped into the vacuoles to cause them to be concentrated, hence having lower water potential than the soil, enabling them to absorb water. Here is a summary of the similarities and differences between these three modes of movement of substances.
Now let's move on to biological molecules. Basic units are absorbed by cells and are the basic building blocks of life. There are three biological molecules and the elements in them that you have to know. The molecules are carbohydrates, fats, and proteins. Carbon, hydrogen, and oxygen are present in all three molecules, while protein has additional nitrogen compared to the other two groups, and sometimes sulfur might also be present in protein. Here's the list of basic units found in their respective biological molecules. The most commonly seen basic unit of carbohydrate is glucose, and two molecules of glucose will make up maltose, while glucose and another basic molecule will bond to form sucrose. Fats are made up of fatty acids and glycerol, while proteins are chains of amino acids. When many basic units bond together, they will form macromolecules. Starch, glycogen, and cellulose are formed by chains of glucose. Fats are formed by one glycerol molecule bonded to three chains of fatty acids. A chain of amino acids forms a polypeptide, which will fold or combine with more chains to become a protein. Individually, they serve their respective functions. Carbohydrates or glucose are sources of immediate energy, while fats are for energy storage and are important for heat insulation. Proteins function in growth and repair, as well as forming enzymes. Lastly, you will need to know the food tests used to identify the presence of each nutrient. Just remember Benedict's test only works when there is a hot water bath and produces other colored PPT as well, when concentrations are lower. Iodine can be colorless when it is very diluted. One of the important molecules which proteins fold into are enzymes. Enzymes are biological catalysts which speed up chemical reactions. They can either synthesize or break down their substrates, which are specific. The poor analogy used is the lock and key hypothesis, where the enzyme is the lock because it has an active site that is like a keyhole. The specific substrate is like the key to its respective lock. We say that the active site is complementary in shape to the substrate molecule, so only the substrate can fit into the active site, just like how a specific key fits a specific lock that is complementary to it. After the substrate binds to the active site, the enzyme speeds up the reaction by lowering the activation energy. All chemical reactions need to reach a certain level of energy before they can take place. The enzyme will form an enzyme-substrate complex with the substrate and lower the activation energy for the reaction to occur. Another property of enzymes is that they remain chemically unchanged at the end of the reaction. Therefore, only a small amount of enzymes is needed since they can catalyze many identical reactions. Since enzymes are made of protein, for your O-level syllabus, they are sensitive to changes in temperature and pH. At low temperatures, enzymes are inactive. Their activity increases as temperature rises because they have higher kinetic energy, increasing the chance of binding with a substrate to form an enzyme-substrate ES complex. This continues until the optimum temperature of the enzyme, where the rate of reaction is the fastest. Different enzymes have different optimum temperatures. After this point, the enzyme will start to denature, resulting in the loss of the active site, and the substrate can no longer bind to it, causing the rate of reaction to decrease. For pH, it is similar. Each enzyme has an optimum pH. Different enzymes have different optimum pH values. For this enzymes, it is pH 7, while pepsin in your stomach has an optimum pH of 2. Both an increase and a decrease in pH from the optimum will cause the enzyme to denature.